So hello everyone, I'm Rosemary from Molecular Cloud, and today we are interviewing iGEM Groningen. So would you like to tell us more about your team and introduce yourselves?
Yeah, sure. So my name is Charita Sarkar, and here is my teammate Ilya Ivanov, and we are from Team Root Patch, iGEM Groningen 2020. Our project is about creating environmentally friendly specific solution to the potato root cyst nematodes. Well, to start with, this problem is not only, I think, a very local problem, but the cyst nematodes have been a problem for this potato industry in many parts of the world. And in Europe itself, close to 460 million euros are worth of crop is lost every year because of this issue. And the existing solutions that we have is either using very harsh nematicides, which also affect the good ones, which is necessary for the soil biodiversity, so it harms the rhizosphere. And the other last moment solutions are flooding the entire field, which again harms the quality of the soil as well as it's not actually a very specific solution, it's inefficient. So which is why we wanted to come up with something that is specific to the harmful nematodes. And root patch is basically a protective biofilm around the soil in the root. And the genetically engineered bacteria that is present in the roots secrete something called NLP, neuropeptide-like proteins, which is chemically identical to the neuropeptides that are secreted by the nematode's own neuronal system, and that kind of confuses them, and that results in a repulsive behavior that drives them away from the root. Instead of killing every other organism or the beneficial ones, it just targets just specifically the one that's harming the crop. And our target this year is one specific type of nematode called the Globodera pallida.
That's incredibly innovative. I think it's great that as you said, instead of using, some sort of pesticide or trying to get rid of the actual nematode, you're just modifying the behavior to stay away from the actual root of the crop. That's incredible.
Yeah, that is what we were pursuing as well, because many current solutions rely on using some forms of toxins, and the toxins can have unpredictable behavior to effect towards other nematode species or even other microorganisms. We can't confirm 100% that our solution would not, but at least it's a molecule that is native to the organism, so it's not some chemical that is brought into the environment and is not native there.
I see. And have you explored other beneficial ecological effects that could result from changing the nematode's behavior?
While this is the main benefit that we were going for, but again, one question that keeps arising on the flip side of the coin is that if it would result in it spreading in other parts of the plant or maybe affecting some other organism. So this is very specific to the chemical that's released by the nematode, so which is why it will not harm any other soil organism, or in fact any other nematodes as well, the beneficial ones. This is very specific to Globodera pallida. And second of all, we have like really good kill switch systems. I mean, the whole project is conceptual. We have not been able to enter the wet lab because of obvious reasons, the pandemic. But conceptually, we also have very strong kill switches to prevent the spread of this biofilm into other parts of the plants or anywhere else in the soil, in fact, making it dependent on like the root, what should I call it? Substances which are released by the potato cultivar itself, maybe like strigolactone or solanine, chemicals like these, which are only secreted by the potato root. So which is why in any case, the biofilm tries to spread into any other part of the plant or soil, because it's dependent on these chemicals, it will not survive. So we also have very sturdy kill switches to tackle these kind of problems.
Excellent. So does that have to do with the cyst clearance? Are the
Uh,
kill switches what you use for the cyst clearance, or is it mainly the function of the APG to enhance the cyst clearance from the soil?
this is not exactly for cyst clearance. It's mostly to ensure, like this is a part of the biosafety, that
I see.
Yeah.
that the biofilm that we are using is not present in anywhere else but the root. I mean, it's supposed to be specific to one part of the plant. So you don't want to find it on the stem or the leaves, or in fact on any other part which is not close to the root. So which is why, to make it stick to where it's supposed to work and to make it more effective, I mean, I think the kill switch is going to do that.
Yeah, I guess you could call it a safety mechanism or a safety switch, which prevents the spread, because at least the soil in the agricultural fields, we can kind of predict it, even though not really, but we know what it is like. But what if it spreads potentially to soils that are not in the field, like nearby forests or grasslands, something like this, and the ecosystem there is quite different, and we have no clue what will turn out to happen. So we are designing this safety mechanism to ensure it stays where it's needed.
I see. Okay, I see. And how is your team currently carrying out your research? So are you in the lab? Are you in the field? Are you modeling? Maybe because of the COVID limitations.
We, you want to answer?
I don't know, please go ahead. I mean, it's
Right.
all the same. We're the same team.
Right. So we weren't able to enter the labs due to the pandemic, of course. But we didn't know it initially, so we did have to restructure our approach to this problem. We still design, yeah, with the models, we aim to identify in some of the aspects that could be limiting factors or certain challenges in implementing it into real life conditions, let's say. Even though, of course, our models are not as accurate and deliberate as the real life models out there, but we're trying to simulate as best as possible. So that's where we are at. And also, I'm not sure if you heard or not, but we're trying to reach out to companies and laboratories in the Netherlands, but also outside, to either carry out some of the experimental work there at their labs, or maybe cooperate so that they would do some of the experiments for us.
Excellent. And if you took a more corporate route, more industrial route, what might that application look like? A mass study in a field to kind of apply your idea.
Well, at the beginning of the year, when situations were a little better, we had members of our team had visited potato farms. Actually, like two of our members had their extended relatives working in the relevant field, so we had the good opportunity to go and visit these farms. And in the middle, we couldn't really move much. But again, recently, like last to last week, probably, we had the fortune to go visit the Agrico lab. I think Ilya was there, so he would be able to tell about it better.
Yeah, I was there. It was really nice. Big thanks to Sevka, CEO, and showing us the Agrico facility and having a wonderful conversation. He also informed us a lot about what are the possibilities of it reaching market as a product, what could be the possible hurdles. We are aware that in the EU specifically, the GMO plants and even more the GMO bacteria and organisms are very unlikely to reach the market, just due to the public opinion and the amount of regulations and things you have to go through in order to make it happen. Nobody will go that far. But it also has certain advantages compared to designing GMO plants. For instance, when you design a product with bacterium, that is final and you can apply it to any kind of variety or cultivar of potato, whereas with if you genetically engineer potato, once that cultivar is no more needed, you have to go through the whole process again. So we're even though we are asked this question quite often, how do you plan for it to reach the market by sponsors, etc. We we don't invest such a big effort into that because we know that it's a long and difficult process to reach that. So we're just considering different alternatives.
Okay. Do you think that this application could be a model for applications to other crops as well and have an even greater impact with pesticide control? I mean, pest control.
Definitely, definitely. This field of this NLPs, neuropeptide-like proteins that we are using, it's relatively under-investigated at the moment, but hopefully more research will come into this area. And as you know, many plants are affected by nematodes. Those are tomatoes, beetroot, onions, even strawberries. I think it's all kinds of plants. And as more of these NLPs will be identified, considering that we're able to do the proof of concept for this technology, in principle, the same could be done with either with other NLPs, other nematodes, other environments, bacteria. So what we see potential in is the approach, this innovative approach of for crop protection in general.
Awesome. That's very innovative. And, you know, I agree. I think it definitely could have a greater impact, not just on plant roots, but across all types of crops, as you've mentioned. Is there anything else that you would like to share with our audience about your team or your project?
Well, I think we have a really cool outreach program as well. Initially, we started with a MOOC, a massive online open course. And now we have this kind of series of short lessons for basically anybody who wants to educate themselves more on the topic of genetically engineered organisms. But the target is mainly high school students. And this is, I think, a great step when we want to influence the next generation of scientists into thinking that genetically modified organisms might not be as bad as we think they are, because a lot of people have this kind of prejudice in their mind against these things, and which is why, like Ilya mentioned, the public opinion massively influences the decision whether a product is considered to be ready to launch into the market. And ultimately, then the goal of doing this research is so that somehow we can reach out to the public and this product serves them in a better way. We can create more food and in the process generate less wastage, or I mean lessen the amount of crops that we lose each year to these kind of pests. So I mean, I think that outreach program is also a great step forward to changing or molding the public opinion in a positive direction.
Awesome. Very, very nice. I'm glad that you guys have implemented that problem. I think it will definitely be beneficial to the public and educating them and giving them more insight into what the benefits that can come from genetically engineering certain organisms. Well, if there's nothing else you'd like to share with our audience.
Yeah. I don't know if you would include our email or some of the social media accounts, but if you do, you're all welcome to subscribe and follow our progress and what we are doing next. And of course, contact if you have any questions, and we're always open to any collaborations, if you have any advice, suggestions, or anything.
Okay, well, thank you so much for meeting with me. And just for our audience, just a reminder, iGEM Groningen is participating in our Molecular Cloud and GemScript sponsorship competition. So please go out there and vote for them and their project.
Thank you so much for having us. This was a very fun and good way to interact with Molecular Cloud, and we are so glad we did this.
Oh, no problem. It's our pleasure. Take care. Thank you so much.
Welcome.
Bye
Bye.
bye.
You, bye.